Okay, so a big topic, and this leads into a major area right now. We have so many new drugs coming along, and we have patients who have used and unfortunately are relapsing after using some of the recent novel therapies. And so we now have a challenge where patients have become refractory to the thalidomide and Revlimid and pomalidomide class, the IMiD class, and then also refractory to Velcade and carfilzomib, the proteasome inhibitor class. So the challenge of double refractory disease was discussed at this summit by Dr. Mary V. Mateos from the Spanish team. And so the current definition of this double refractory is, those who are refractory to the bortezomib class and the lenalidomide. And then also the second element where there's a failure to achieve at least an MR and progression on current therapy or within 60 days. And so the question that Mary V asked, which I thought was really pertinent, is, what do we truly have to offer this double refractory population right now? And so I'll just take you through some of the more recent information that we had from ASCO. So obviously there was a lot of excitement about the Eloquent True trial, which is obviously the assessment of the monoclonal antibody elotuzumab with or without lenalidomide and dex. And so, but important to point out here is that this was not in patients with relapse refractory disease. It was in earlier disease. And so this phase 3 trial in patients with 1 to 3 relapses showed substantial benefit both in PFS and survival in this population. And so a lot of excitement about the added value of the elotuzumab, and especially this point which has been discussed, that the patients are having a more sustained benefit, and so longer PFS at the longer time points, which was another aspect for discussion. But relevant to the double refractory, I think that there was truly much more significant excitement about the anti-CD38 patients, the SAR compound, but particularly the new data that was presented on daratumumab. And this was really where we were looking at this double refractory population and really seeing some significant results. And the results were presented in these different categories here, looking at patients who had had different combinations of prior therapies. And so, as was emphasized by Mary V when she was reviewing this, it really impressive to see as a single agent the remissions and the survival in this double refractory population. And so if we go a little bit further, does this type of efficacy represent a model of what we're trying to achieve? I think that we had a lot of discussion about how do we evaluate benefit in this double refractory setting. We talked about the results with panobinostat. And so, but here we have, I would have to say, a more decisive benefit. But is this the kind of benefit that we're looking at to encompass a true unmet medical need? And so, number one, do you agree that this is really an important drug in this setting? And is this the kind of thing that we really need to be looking for? Paul?
Well, obviously, both Dara and Elo are very near and dear to our hearts, because the Elo story begins obviously before Dara, and I think what's particularly attractive about Elo is to see the success of a rational translational effort in which, when we used elotuzumab as a monotherapy, the best we saw in relapsed refractory disease was just stability. When we put it into overdrive by combining it with immunomodulatory treatment, which was informed by laboratory observation, to see it translate into this trial result, I think was very gratifying. And I think for patients it's incredibly important because, as you pointed out so nicely, Brian, the clinical benefit seen recognizing that the overall result was 4.5 months or so. The clinical benefit in particular groups was particularly striking.
Yeah.
And what I think jumped out at me, which I think is very important for patients to appreciate, is that for those patients who achieved complete response,
Right.
if you got the 3 drugs versus the two, there was a substantial difference in terms of clinical benefit in favor of the 3 for
Right.
duration of disease control and progression-free survival. So that was particularly striking. What was also very striking was the clinical benefit seen in the adverse cytogenetics, very much mirroring what we would expect. And then the secondary endpoints of response difference clearly supporting it. So a very positive trial. I think elotuzumab will be the first antibody approved on that basis.
I've given you one scenario here, but this other scenario is equally important, where you have an added benefit in an earlier disease setting, in a randomized trial setting. And a key point in certainly the way that I'm looking at that is that we have an advantage with a new class of agent. So an immunomodulatory agent.
Well, yes, exactly. As an antibody, yes, exactly. And I think the DARA data are very striking. And again, they reflect what was seen in the phase 1, but recognizing the phase 1 populations highly selected. This phase 2 experience that was multi-center provides an accelerated approval platform for DARA, I think.
Right.
You mentioned panobinostat, and I think it's very interesting because, again, if you start to look at subgroups, when you look at the panobinostat data and realize what the FDA was so constructive in dissecting out from the panobinostat study was that those patients in whom immunomodulation and bortezomib-based therapy had failed them, that was the group in which the progression-free survival was most striking. And actually it was 6 months. So I think these studies tell us a number of things. I think they tell us that, with new mechanisms of action, that's where the sort of breakthroughs are coming. And I think that daratumumab's single agent efficacy is striking. We know that when you partner it with other drugs, there's even more excitement to come. And ELO has already established that and will be kind of first through the box, as it were, out of the box in that regard. But DARA will be hot on its heels, and I also would predict that isatuximab, or isatuximab is the new name for SAR. I'm actually glad because
it's got a name.
Well, we used to call it DAR and SAR, didn't we?
Yeah, DAR and SAR.
And I think now it's daratumumab.
Think of Isa wearing a tuxedo.
I love it.
Isatuximab.
And it makes great sense. But I think isatuximab will also emerge in that same context. So I think very exciting, Brian. Very good news.
So what's your reaction to this? Obviously, I've given you one example here with a single agent and the unmet need, but also the combo earlier.
I mean, I think it's great. I mean, I agree with what Paul's saying. I think there's kind of 2 ways I look at this. The phrase unmet medical need has a very defined purpose, right? This is looking at a regulatory pathway for a drug to be approved, and those are the rules of the game. That's the way the game gets played, and we want to play that so that we can jump through the hoops to make sure we have better access for better agents for our patients. There's no doubt. How that's exactly defined, I think the phrase double refractory is going to have to go away eventually, because now it's very complex. There's all these different agents, and double means something to somebody else as to even if we just confine it to the United States, because really the double refractory unmet medical need was being met by some degree with carfilzomib and pomalidomide. So in fact now we have, as you showed with the DARA study, as we can with isatuximab, we have these quadruple refractory like patients. So there's the regulatory way, and then there's just the way of thinking about what is going to be good ultimately for our patients. And I think that's what we're excited about, is that we have a different mechanism. We can approach the disease in a different way. And maybe one of the things we haven't had a chance to say yet is with these monoclonal antibodies, in particular elotuzumab and the anti-CD38s, not that every drug has some risk, obviously, and there's always adverse events or side effects from any of them. The kinds of side effects that we see from these drugs are not typical of the ones we've seen with the other drugs. They don't have what I call overlapping toxicity, so the ability to combine them is enhanced. Not just because biologically they work together, but we can combine them. And now the data that we're seeing of combining DARA with lenalidomide or isatuximab with bortezomib, these kinds of studies are very encouraging to us to be able to bring us further along the way.
And Joe, to your point, what was very nice in the ELO trial was to see that the the efficacy signal from both over the age of 65 and under the age of 65 was the same. And what was also particularly striking was actually if you drill down on the adverse event data, very similar. So certainly speaking for elotuzumab, its tolerability is quite striking and the rate of infusional reactions is really quite low. So I think I completely agree with you. I think it's extremely exciting and great for patients. In terms of the new terminology, Brian, I think we have to be a little careful because I think that double refractory means clearly something in terms of a regulatory pathway. And I think we have to very much engage here with our regulatory partners and our pharma partners to make sure that there is a unanimity of understanding. Because I thought what was one of the most encouraging things from the summit, and again, enormous credit to you for organizing it. I think it's one of the highlights of the year, in my view, is that we get people together where we can have these discussions. And I think the discussion that both Joe and I were at yesterday, I thought was very fruitful because we recognize that double refractory means different things in different countries. And I think we're going to probably have terminology which is broad and then drills down. So we're going to have sort of essentially, are you refractory to the IMiD class, are you refractory to the proteasome inhibitor class, and which agents, recognizing that IMiDs are different.
They're not the same drugs.
Potencies are radically different. And also, of course, in the proteasome inhibitor world, it's even more stark because they are clearly different drugs. So I think that we'll have that. Then I think there's going to be the next category. And we discussed this at the session this morning, which is now we're going to have refractory to next generation new mechanisms of action. So as we go forward, how are we going to address...
The complexity becomes mind-boggling.
Right, and actually we're going to have patients who are resistant to monoclonal antibodies. So that, and to me, if you have a single agent that is able to overcome, the so-called quadruple refractory trials where patients have unfortunately had IMiDs, proteasome inhibitors, and second generation carfilzomib pomalidomide fail them. If a drug has a signal in that population, that's a very provocative sign. And I think we'll have antibodies and HDACs coming into that same space.
And that's part of the element of this question, is that if you have something that's working in this setting, that's very important.
Right.
So I think this is kind of a paradox. So as we are moving into the future when drugs are better and better, it's getting harder and harder to get them approved because the competition is tighter. And I agree with everything you say. The rules are set up by the federal agencies that regulate and approve drugs, and we have to have rules. But on the flip side now, if you look at the prevalence of patients with this disease, 5 years ago, I think there were 55,000 patients living with myeloma in America. I think this year there are 86,000. And the projection is in 5 years there will be maybe close to 100,000. And this is because there is no cure for the disease that can be guaranteed. So clearly there is an unmet need to develop an established cure. And now we are talking about the problem of getting drugs approved. So this is the ethical dilemma.
And so, as you know, we're going to be leading into this in just a moment. I was making note of these triple and 4 and 5 drug combos that you were ending up discussing there, which have significant financial implications.
Brian, if I may, one sort of last point on this is I do think, and particularly speaking for the FDA, they are very, very, I think, engaged in the myeloma space, and I think they've been remarkable partners. I mean, there's no question. And what I saw them do in terms of helping with panobinostat was unprecedented. I mean, they stepped in and sort of said, we see you've got a positive signal, let's sort this out, despite an ODAC that, on the face of it looked like, no way. They actually went in and said, wait a second, here we are. And I was very struck by that they basically said, you've got a double refractory population and you've got a novel mechanism of action. So they drilled their approval down into that. And I wonder if that's what we're going to see, that essentially what they'll say is, you can have double refractory, we're not going to raise the bar too high because it's going to become impossible. What we're going to say is, if you've got a novel mechanism of action, are you offering the patient something that's completely different? And I think that's going to be a very exciting new avenue for drug approval.
Absolutely, absolutely. So one aspect of this I just want to touch on before we move forward is we've touched on the elotuzumab, which we're obviously excited about, the daratumumab and the SAR. I'm going to stick with the SAR for now.
You can't think of Issa wearing that tuxedo?
Beyond that, I've given another 2 or 3 things there which are exciting, and we heard lots of different ideas about what would be the most important next thing. So maybe each of you could give me, beyond the top 3, what are the next couple of things that you view as the most exciting? So we'll maybe start here. Ola, what do you think? Beyond the top 3, I've given you some examples there, but what do you think is the next couple?
So can I only pick one, or
2? No, we'll give you, let's start with 2. Let's start with 2.
well, I mean, I think I think that because drug development is complicated, I think we have to risk spread for sure. So what we are working on is CAR T cells.
Okay.
We will probably have CAR T cells coming to clinic within 12 to 18 months at our institution.
No, the recent results were with the CAR T 19
Correct.
population. And so is this what you're talking about, or are you talking about the other?
We have other targets that are tumor cell specific. And we have also manipulated them in other ways.
So just so that we're clear, so obviously in the CAR T19, this is something that affects about 4 or 5% of myelomas, which are typically CD19 negative, but 4 or 5% are CD19 positive, and so the activated T cell therapy has been actually remarkably effective in some of those patients. But so what percentage of the patients might be affected by your newer therapies that you're describing?
Are we talking about potential candidates who could be attacked? So if we were to screen patients for the antigens that we are planning on targeting, probably 60 to 70% of the patients could have those targets. But we need to do a little bit more work. My second choice, and I'm not kind of ranking them here, but you said I could pick 2. So I do think that it's also exciting, beyond just looking for new targets, to also to look how you can go after a target that we know works. And one of those is to inhibit the proteasome.
Okay.
Because we know plasma cells are very dependent on the proteasome. We have been working, and I've been working together with Paul, we are working on a joint project with early drug development, inhibiting upstream of the 20S proteasomes. We are blocking the 19S proteasome unit. Okay. And that seems to work even in the lab when the tumor cells have been exposed to the carfilzomib and Velcade. When we give these drugs, they still die. Okay. So now we are using this in the clinic. So we have a new trial. I'm very excited about that trial.
Okay.
If I may, just because it bounces off,
so,
and I apologise, Joe, for jumping in.
So Paul, you're next.
Well, no, only because we've got this super partnership with Ola on this DUB inhibitor concept, and I think it's very exciting. I think, as we think about the other that you have in this category, I completely agree with Ola that there is more to be exploited from proteasome inhibition upstream, and very interestingly, there is the recognition that our current armamentarium of proteasome inhibitors primarily target the beta-5 subunit, be it bortezomib, be it ixazomib, be it carfilzomib or aprozomib. There is a new game in town, relatively new, arguably a little bit old. It's coming through slowly but surely, and that's marizumab. Marizumab hits beta-5, beta-1, and beta-2. And what's so interesting is that when you actually model this, the proteasomes upregulate beta-1 and beta-2 to counteract beta-5 inhibition. Well, this molecule hits all 3, and it's in the beta-lactone class. And again, informed from the laboratory, we've partnered it with pomalidomide. And in the laboratory, it's the most powerful of the IMID proteasome inhibition synergy that we've seen. And clinically, it's funny, I was just with Andrew Spencer, who's a part of our trial, and everybody in the trial, Ashraf Badros is part of it, there's a great team. Everyone's stunned. We're basically seeing with marizumab POM response rates that are, every single patient treated so far has responded to this platform. And so I think there's a lot more that we can exploit from the proteasome inhibitor pathway. And then, if I may, just for my second choice, I would simply say that the other area that we're particularly intrigued by is the checkpoint inhibition story. And I think, good for you, Brian, for having it up here. And I think we heard so nicely at the summit from everyone about the quote-unquote Achilles heel of immunotherapy in myeloma. And I think checkpoint inhibition, not on its own, but in combination, is going to be very exciting. And I'm sure, Joe, you can comment more on that.
Yeah, well, we've been doing some work with checkpoint inhibitors as well. I tend to agree with you. I think on their own in myeloma, they may not be that potent. You guys haven't left me much left. Actually, no, there is a lot left. So just following the proteasome inhibitor theme, let me just also remind us, because they're not on the list, the two drugs that you mentioned, they may not be scientifically a massive step forward, but for patients, a huge convenience step forward have been the oral proteasome inhibitors.
That's very true.
Yes.
So ixazomib, which is really an oral version of bortezomib, and oprozomib, which is really an oral version of carfilzomib. Although I must say, in one of the studies that we did, we took patients who were refractory to carfilzomib and treated them with oprozomib and saw a signal. So I'm not sure oprozomib is exactly just oral carfilzomib. And because it's there on the list, the selinexor is a drug that has been a bit of a challenge for some patients, for sure, especially in lymphomas. But I think we're learning ways to try and overcome that, some of the GI problems that they've had.
And so in combination with Dex.
Absolutely. And then lastly, just on the monoclonal antibody theme, the other target that is quite prolific, maybe not as prolific as CD38, but CD138. And so now doing some early work with some of the anti-CD138 inhibitors is showing promise. So I think the message really is we've got a lot of promising targets and agents at various stages of development. And I suspect if we have this conversation again in a year from now, your list is just going to get longer and longer, Brian.
Well, I think it is. And I think one last comment, if I may, Brian. Just as we saw the ultimately, the getting across the goal line with panabinostat, we've also been very impressed that there's a next generation of HDAC inhibitors that are more selective, better tolerated, and arguably potentially more active. And the other trial I'm very excited about, in combination with pomalidomide, has been our experience with the AC1215 and POM, where certainly in our own experience, for our own cohort of patients, the responses have been remarkable. And what's really interesting is the tolerability of the IMiD plus the HDAC has been striking, whereas some of the more challenging side effects that we've seen from combining the HDAC platform with proteasome inhibition, which has been so synergistic but so challenging from a tolerability point of view. When you partner these drugs with the IMiDs, it's fascinating. We're seeing excellent tolerability from an all-oral regimen, and in our own experience, particularly with the 1215 POM platform, the responses have been dramatic. So I think you're absolutely right, Joe. I think there's a lot coming. And I think the challenge for us as investigators is how on earth we make sense of all of this. But I would say one thing, it's clearly not a zero-sum game. I mean, we're very clear about that. Patients often ask me that, where do we go? We need all of this, because the reality is I have patients in whom daratumumab fails them. I have patients in whom elotuzumab fails them. And I'm sure, Joe, you've had the experience with SAR.
Clearly an unmet need. Yes.
Absolutely.
So maybe we'll not all answer this question, but another topic that came up just to touch on is that in this setting we have increasingly patients who've gone through these different novel therapies but have not had full exposure to the alkylators. And so there was discussion at the summit here about alkylator therapy and the use of stem cells transplant in more of the salvage setting, and patients who maybe have had a lot of novel therapies but actually never did have a stem cell transplant. So do you look at that as one of the options possibly in this more relapse situation?
For sure. I had the privilege of being involved with two of those studies where we looked at using a salvage second transplant. So that's another, sort of a theme in myeloma, of course, is that you can go back to a drug that's been used before, especially if it worked before and had a period of time. So you're right, there are a lot of patients now, especially older patients, for example, in whom we're not giving the melphalan early on. A lot of these patients end up going through a lot of novel therapies, as do younger ones who may avoid transplant or have a single transplant. That I do look at this list. I mean, I tell patients it's not a game, right? But I want every card available to play. I want to play the best card for my patient, but I also recognize that I have other cards to play. And in some patients, I had a patient last week in clinic, he went 4.5 years after his first transplant with no maintenance therapy. You know, 5 years ago we weren't using it as much. And he said, well, you know, we banked enough stem cells, and can I have a second transplant? And why not? You know, because it's likely that he'll at least get 50, if not 70% of the time that he got after the first one. So in simple answer, I would say yes, Brian, it is an option. I think there are challenges in using alkylators, challenges using them a lot up front because they can be tough on the bone marrow.
Right.
But as an option later, especially in combination, be it cyclophosphamide or melphalan, I think these are agents that we will still be able to use. Like Paul said, we don't want to give up anything we've had. We want every option on the table because more options means better options for our patients.
So talking about having every option on the table.
But Brian, if I may, just as I'm so sorry.
I know where you're going.
No, because we're almost getting there. But I just want the reason I wanted to just chime in very quickly on that, because it's something again that Ola and I are working together on, is the recognition that there are new chemotherapeutics that have the opportunity to be less toxic, especially in terms of stromal injury and stem cell damage. And they're teaching us some very interesting things. And at the EHA meeting is being presented data from an international study group looking at a new melphalan derivative called melflufen. And I have to say, the results are striking, to your point, that you can revisit. And one can argue, have you reset biology of disease? Because what you're doing is you're treating with novel biological agents, and then you can come in with a novel cytotoxic and see responses. And I think it's very exciting, and it echoes what you're saying about the second transplants. Yeah.